Hello everyone. In this lecture, we are going to discuss about the cellular basis of biochemistry, that is cells and their organelles and their function. Cells are the essence of life. All living beings are made up of cells. A cell is the smallest unit of life within an organism. This means that they perform basic functions of life, including feeding, socializing, and reproduction. It's like a tiny creature. Most organisms are multicellular organisms, but there are also single-celled or unicellular organisms, which can only be seen through a microscope. But now the point is that what the cells are made up of. Cells of animal and plants both are composed of three main elements: the cell membrane, cytoplasm, and the nucleus. But before discussing the individual organism, let's discuss that how a basic unit of a cell is synthesized. Imagine we have a beautiful article with us. What if we want to make a copy of it? The best way would be to make the use of a cast or a mold. Similar strategies are used within ourselves. The most important task for a cell is to synthesize proteins which are based on the codes of DNA. But how these codes are read? What's the process that helps to decode this language? And are the proteins synthesized merely by the sequence of nucleotide A, T, C, and G? The answer of all these questions lie in the central dogma of life. But what exactly is the central dogma? Well, one makes copies of itself by the process of replication. So this DNA, which is going to make its copy by the process of replication, now there is a post-replication step in every cell. So the code of the DNA is cracked and then transcribed into a messenger RNA. This messenger RNA is now read, or this code is translated into the protein. This interesting concept is basically the central dogma of life. Now let's discuss about the individual organelles of a cell. So if we are talking about before discussing, now let's discuss about the individual. Now let's discuss about the types or different categories of the cells. There are two main types of the cells, animal cells and plant cells. Both cells are made up of quite different. There are two main categories of the cells, the prokaryotic cells or the eukaryotic cell. There are two main categories of the cell, the prokaryotic cells or the eukaryotic cells. Pro means false, means the organisms which have a false nucleus or which do not have a completely developed nucleus. And the example of these type of organisms are bacteria, which have no membrane-bound nucleus, and the nucleic acid is usually found in loops in the cytoplasm, and they are usually smaller than the eukaryotes and having fewer organelles than a eukaryotic cells. On the other hand, eukaryotic cells are larger and have a membrane-bound nucleus where whole DNA is located, and it contains more organelles than a prokaryotic cells. All organisms except bacteria, blue-green algae, and cyanobacteria, they all are eukaryotes. So this is the basic difference in a prokaryotic cell and a eukaryotic cell. Prokaryotic cells are organisms including bacteria and cyanobacteria. However, the complex organism, all the complex organism, including plants, fungi, and animals, they are included in the eukaryotic cells, or they all are eukaryotic organisms. Prokaryotic cells lack the nucleus and other membrane-bound organelles, and they have a single chromosome in which DNA is associated with known histone proteins. On the other hand, eukaryotic cells contain nucleus and membrane-bound organelles, and it contains several chromosomes. Usually, prokaryotes exist as a single virtually identical cells like bacteria, or in other cases, it could be associated with other cells. On the other hand, eukaryotic cell can specialize for certain function, multicellular organism and multicellular organs and organisms. Cellular respiration in a prokaryotic cell is occurred in mesosome. However, in eukaryotic cells, cellular respiration occurs in mitochondria, ribosome, a very important organelle. The ribosomal subunit are found to be as 30S and 50S ribosomal subunit in a prokaryotic cells, which are found to be as 40S and 60S ribosomal subunits in eukaryotic cells. And the process of photosynthesis in a prokaryotic cell occurs in chlorophyll-located region, while the photosynthesis occurs completely in the chloroplast in a eukaryotic cell. The prokaryotic cell contains cell wall, and the eukaryotic cells, in eukaryotic cell wall is present in plants and fungi only. So what are the eukaryotic cells? Eukaryotic cells, they are 100 times larger and much more complex than a prokaryotic cell. They contain organelles to support different cellular function, from digestion to generation of energy and storage of nutrients. A eukaryotic cell contains extra chromosomal DNA as well, which exists in mitochondria and chloroplast in case of the plant cell. And they are differentiated to different cell types. And these cell types could be like neurons, they could be like muscle fibers, and so on, which are specified for specific functions. However, a prokaryotic cells, they are small and simple. They grow and divide fast. They can adapt in rapid environmental changes. There is a single compartment with DNA and other biomolecules in a prokaryotic cells which exist together in a well-organized way. The extra chromosomal DNA exists in the form of plasmid in a prokaryotic cell. So this is the model of a eukaryotic cell. Here you can see the nucleolus, the nucleus, ribosomes, vesicle, rough endoplasmic reticulum, the Golgi apparatus, vacuoles, cytoplasm. This is the cytoplasm, lysosome, centrioles, . Animals and plants, both are made up of the cells. They are quite different. The plant cell have an outer thick wall that protect the plant and keep it upright. They also have chloroplast, organelle, whose function is basically to make food. There are many types of cells with different shapes and function, such as neuron, which is responsible for transmitting information, and muscle fiber, which is capable for contraction. Similar cells are grouped together in a tissue in order to perform a specified task. For example, muscle fiber is responsible for contraction. The union of tissues build up the organs which are specified for their function. The heart uses muscle fibers for the contraction and to pump the blood. Organs are then grouped to form an organ system with the common mission. The circulating system is responsible for distributing nutrients throughout the body and thanks to the heart. Here you can see variety of organelles in a eukaryotic cells like the nucleolus, nucleus, ribosome, vesicle, rough endoplasmic reticulum, Golgi apparatus, smooth endoplasmic reticulum. Here is the smooth endoplasmic reticulum. Okay, other than this, there is a cytoskeletal structure or the cytoskeleton. Here you can see the structure of mitochondria, vacuoles, which are present over here. These spindle-shaped centrioles, and so many organelles within a eukaryotic cells. And some are membrane bound while the other are non-membrane bound. Okay, the cell wall and extracellular matrix. Bacterial cell wall is made up of carbohydrate, which is covering the outside of the cell membrane. For example, gram-positive bacteria, they have simpler but a thick wall of a peptidoglycan in order to protect the cell. On the other hand, gram-negative bacteria have less peptidoglycan or a thin layer of peptidoglycan, but it is more complex in structure with periplasmic space and lipopolysaccharides. Plant cells also use cell wall to stabilize their structure. However, the animal cell have extracellular or outside the cell, there is a matrix in animal cell instead, and they are more dynamic, being formed or disorganized quickly. So what's the basic difference in animal cell and a plant cell? As we have discussed earlier that both animal and plant cell, they both are made up of the cells, but these cells are quite different in a way. Both are eukaryotic, contain organelles for specific function. They contain mitochondria and contain cytoskeletal proteins to hold up shape and the structure of a cell. They contain nucleolus and nucleus for the ribosomal RNA synthesis and contain both rough endoplasmic reticulum. Okay, so rough endoplasmic reticulum is present in both animal as well as in plant cell. Animal cells have the ability of differentiation. Specific cell types are for a specified biological function. For example, different cells are for production and the secretion of specific enzymes and hormones. So, animal and plant cells, how they are differ from each other. Plant cells and fungal cells, they have a proper cell wall, which is made up of carbohydrate. Plant cells have mitochondria and chloroplast is only found in plant cells. However, animal cells contain mitochondria. Vacuoles and storage granules in the plant cell are responsible for water control and the storage of the nutrients. On the other hand, animal cells have lysosomes for the digestion, microtubules, microfilaments, and centrioles for intracellular transport and cellular division. So here you can see the structure of the vacuole that contains variety of digestive enzymes. So now let's discuss about the chemical composition of the membrane or plasma membrane. Plasma membrane is composed of lipids, protein, and carbohydrate. All membrane carbohydrates are covalently attached to the protein and lipid molecules. It contains around 43% of the lipid, 49% of the protein, and around 8% of carbohydrate. So, according to the model of the plasma membrane, which is fluid mosaic membrane model, this model suggests that plasma membrane structure and function. Regarding the structure and function, plasma membrane is composed of phospholipid bilayer that contains phospholipid and protein. Plasma membrane is permeable to a variety of molecules, and this permeability occurs through diffusion, osmosis, and the transport by various carrier proteins. So here you can see the structure of plasma membrane, which is a lipid bilayer structure that contains hydrophobic heads and hydrophilic heads and hydrophobic tails, which are embedded inside. This is the structure of lipid bilayer. Here you can see so many intrinsic and extrinsic proteins, a lipid bilayer, and the cholesterol. So the function of plasma membrane is usually to separate internal living cytoplasmic content from external environment of the cell. And the phospholipid bilayer, it contains external surface lined with hydrophilic polar head, cytoplasmic surface lined with the hydrophilic polar head, and the non-polar hydrophobic fatty acid tails sandwiched in between. Here you can see these are the hydrophobic fatty acid tails, and the hydrophilic polar heads are surrounded to the external environment, or they are faced to the external environment. So according to the fluid mosaic model, which was proposed by the Singer and Nicholson in 1972, there are three main components of a plasma membrane. The basic membrane, referred as phospholipid bilayer. It contains protein molecules, which are floated around like iceberg on a sea. The membrane proteins may be peripheral or they could be integral if they are peripheral proteins. So the peripheral proteins are found on the inner membrane surface. However, the integral proteins are partially or wholly embedded in the membrane. Some have carbohydrate, which is attached to the proteins. And the third component is the cholesterol. This is the fluid mosaic model. Here you can see some are intrinsic proteins, some are extrinsic protein. Here is a phospholipid bilayer. Here is the hydrophilic head and the hydrophobic tail, which is on the interior side. So this is the fluid mosaic model. So what is the function of the plasma membrane, or what is the function of the proteins which are embedded or which are present in the membrane? They are working as a channel protein. They are tubular and allow passage of molecules through the membrane. They are working as a carrier protein combined with most of the substances which are going to be transported and assist the passage of molecules through this membrane. They function as cell recognition. They provide unique chemical identification for the cells or a specific ID for the chemicals which have to be transported within the cells, or which have to be releasing out, and which needs to have entry within the cell. These cell recognition proteins help the body to recognize the foreign substance. In such a way, they are also involved in the immunity perspective. On the other hand, there are certain receptor proteins which binds with messenger molecules and causes cells to respond to the message. Moreover, this plasma protein or the membrane protein contains variety of enzymatic function or possess variety of enzymatic function. They carry out metabolic reactions directly. So plasma membrane is differentially selectively permeable membrane, which allows some material to be passed in and out and inhibits the passage of most of the materials. The passive transport requires no ATP. The transport occurs by either actively or passively. The passive transport required no ATP and the molecules follow concentration gradient. On the other hand, active transport require carrier proteins with the expense of energy in the form of ATP. So what are cellular organelles? Cellular organelles are a specialized subunit within a cell that has a specific function and is usually enclosed within its own lipid bilayer. And these organelles are known as membrane-bound organelles. Organelles are identified by microscopy. There are many types of organelles, particularly in eukaryotic cells. So there are two main types of the cellular organelles. Number one, membrane-bound, while the other one are non-membrane-bound. Rough endoplasmic reticulum, smooth endoplasmic reticulum, mitochondria, Golgi apparatus, and lysosomes. They are among the membrane-bound organelles. On the other hand, non-membrane-bound organelles include centriole, ribosome, and the cytoskeletal structures. Now let's discuss about the nucleus. What is nucleus? The nucleus contains all the activities of the cell and contains genetic information which is then transferred from one cell to another cell. So the nucleus contains most of these cell genes and usually the most conspicuous organelles it is. Nucleus is covered by a nuclear envelope, which encloses the nucleus and separating it from the cytoplasm. The nuclear membrane is a double membrane. Each membrane consists of an individual lipid bilayer. Here you can see the detailed structure of the nucleus that contains chromatin, nucleolus, nuclear envelope, inner membrane, outer membrane, nuclear pores, and so on. So what is the function of the nuclear pore complex? Nuclear pore regulate the entry and exit of the protein molecules and RNA from the nucleus. The shape of the nucleus is particularly maintained by the nuclear lamina, which is composed of proteins. So within the nucleus, DNA and proteins collectively form genetic material, which are referred as chromatin. And these chromatin condenses to form discrete chromosomes. As we know that all the human beings, we have 23 pairs of the chromosome within our cells. So the nucleolus is located within the nucleus and is the site of the synthesis of ribosomal RNA, which is required for the translation protein, step of a protein, and the nucleoplasm. The nucleoplasm of a nucleus contains various enzymes such as DNA polymerase, RNA polymerase for messenger RNA, and transfer RNA synthesis. So what is the function of the nucleus? Nucleus is involved in the DNA replication and RNA transcription, which occurs within the nucleus. Transcription is the first step in the expression of the genetic information. And what is the function of nucleolus? Nucleolus is particularly responsible for the synthesis of ribosomal RNA and ribosomes. It contains RNA polymerase, RNase, ATPase, and other sort of enzymes. Nucleolus is also the major site where ribosomal subunits are assembled. So this is the second organelle, which is known as rough endoplasmic reticulum. Endoplasmic reticulum is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Smooth endoplasmic reticulum is generally used for the creation and storage of lipids and steroids within the rough endoplasmic reticulum. Okay, while smooth endoplasmic reticulum, okay, नहीं इसको छोड़ देते हैं. Okay. Another most important organelle is the endoplasmic reticulum. It spins the entire cell and attached with the numerous ribosomes for the protein translation. This type of endoplasmic reticulum is termed as rough endoplasmic reticulum. Rough endoplasmic reticulum play a significant role in the synthesis of various proteins. The proteins made in the rough endoplasmic reticulum are folded in proper conformation, which may be glycosylated in case of post-translational or in the step of post-translational modification by the addition of sugars, and then they are sorted to various parts of the cells. However, the smooth endoplasmic reticulum is generally used for the creation and storage of the lipids and steroids. So, smooth endoplasmic reticulum produce phospholipids. Smooth endoplasmic reticulum is a depot of the calcium ions as well for cellular signaling. Here you can see the structure of rough endoplasmic reticulum. The next organelle, most important organelle, is the Golgi apparatus. What is the function of Golgi apparatus? Golgi apparatus function as a factory in which proteins received from endoplasmic reticulum, they are further processed and sorted for the transport of their eventual destination. The Golgi apparatus consists of flattened membranous cell like cisternae, which is known as cisternae. And the function is that proteins made up in rough endoplasmic reticulum, they are sorted and processed in the Golgi apparatus. Moreover, glycolipids and sphingomyelin, they are synthesized in the Golgi apparatus. This Here you can see the complete structure of rough endoplasmic reticulum and the Golgi apparatus. They are transported, they are responsible for the synthesis of variety of proteins, and then Golgi apparatus is responsible for sorting and for modifying these proteins and to transport them into their actual destination, ultimate destination. The next cellular organelle is lysosome. A lysosome is a membranous sac for hydrolytic enzymes that can digest macromolecules. It contains vesicles of around 0.5 micron in diameter, which is manufactured in the Golgi apparatus. The size and shape of the lysosome change with the stage of their activity, and the pH within the lysosome is distinctly acidic. Some types of cells can engulf another cell by the phagocytosis. This forms a food vacuole. A lysosome fuses with the food vacuole and digests the other molecule. Lysosomes also use certain enzymes to recycle the cell's own organelles and the macromolecules. And this process is known as autophagy. Lysosome contain variety of enzymes, include proteolytic enzyme. It contains nucleic acid hydrolyzing enzyme, lipid hydrolyzing enzyme, carbohydrate displacing enzymes, and so many enzymes. So the example of proteolytic enzyme is cathepsin, collagenase, and elastase. And the example of nucleic acid hydrolyzing enzyme is ribonucleases and deoxyribonucleases. Moreover, lipases, phospholipases, fatty acylesterases, they are included among the class of lipid hydrolyzing enzymes of lysosomal enzyme. And the carbohydrate splitting enzymes included alpha glucosidase, beta galactosidase, hyaluronidase, and aryl sulfatase. And other than these enzymes, there are acid phosphatases and catalases, which are released and synthesized within the lysosome. So what are the disorders which are associated with the lysosomal hydrolases? Lysosomal hydrolases sent to the cis Golgi are covalently modified by adding mannose-6-phosphate. The mannose-6-phosphate receptor recognizes the labeled hydrolases in these trans Golgi and then move them to the endosomes. The eye cell disease is a genetic defect that produces the enzyme without the mannose-6-phosphate. So there will be no enzyme which will be sent to form endosome for the digestion. Glycosaminoglycans and glycolipids, they are accumulated without proper digestions. So the patients which exhibit psychomotor retardation and skeletal deformities, they die early, usually before the age of 8. Another disorder which was associated with the lysosomal storage disease or known as lysosomal storage disease is called Tay-Sachs disease. There is the absence of a specific lysosomal enzyme that causes accumulation of unwanted cellular material in the brain. For example, mental retardation and blindness resulted from ganglioside GM2 accumulation due to the lack of hexosaminidase A. The lysosome cannot function properly or even break down to release not just digestive enzyme, but also so many acids to kill the cells or to rupture the cells. Another disorder is gout and rheumatoid arthritis. Gout is particularly deposition of uric acid crystals of the joints, often from overconsumption of the meat and then ultimately after excessive purine metabolism. Other rheumatoid factors complexes in the leukocyte, means in white blood cells of the joints. These rupture lysosome to release enzymes that degrade the components of the synovial membrane in the joint and causing the great pain or the severe pain and joint deformities. The next organelle is the mitochondria, which is the energy-making organelle of a cell. So there are two membranes, or you can say it is a double membranous structure. There is an inner membrane and an outer membrane. The inner membrane folds to create the matrix space and form inner membrane space in between inner and outer membrane. The matrix contains enzymes to oxidize the metabolites. This is the main component or the main cellular organelle where cellular respiration takes place. The intermembrane has potential gradient generated by pumps at the inner membrane and energy released from the gradient that produce ATP or energy. This is the major function of the mitochondria, which is termed as, that's the reason this mitochondria is known as the energy storehouse or the energy producing organelle. So the mitochondria contain an outer membrane forming proteins which are only permeable to 5 kilodalton molecule or even less. Other enzymes here facilitate metabolism of lipid for use in the matrix and transport of required proteins in the mitochondria. Mitochondrial matrix, on the other hand, it is the internal space with enzymes for oxidation of the pyruvate and fatty acids and for the citric acid cycle. Here you can see the folds, the cristae, which is represented in the box region. This is the inner membrane space. This inner membrane folded into the numerous cristae to increase total surface area for the electron transport chain or for the cellular respiration. ATP synthase and transporters for bolides are going through this membrane. Here you can see the intermembrane space, which is responsible for the enzyme, or which contains so many enzymes which are required for the production of the ATP, and which use ATP passing out of the matrix to phosphorylate the other nucleotides. So this is the complete structure of the mitochondrion. The outer membrane, the inner membrane, the intermembrane space in between the outer and inner membrane. This is the mitochondrial matrix, and here you can see the pores, which are known as cristae. The next organelle is peroxisome. For the oxidation of fatty acids and toxicants, it contains oxidative enzymes like catalase. In peroxisomes can decompose hydrogen peroxide into the water. These peroxisomes are abundant in hepatocytes, where oxidation of the fatty acids and other organic matters occurs and produce hydrogen peroxide. Lifespan of this peroxisome is short, and the numbers of peroxisomes vary from cell to cell. They replicate like mitochondria, and they are not budded from Golgi apparatus. The enzymes are sent and deposited into the peroxisomes. So now let's have a look on the summary of the whole lecture. Eukaryotic cells are highly organized with membranous organelles, isolated compartments for specific life processes and functions. And the organelles within our cell increases the cell's availability membrane space for numerous enzymatic reactions. The most important organelle, which is known as nucleus, it contains DNA for the gene transcription and the production of multiple proteins, which including RNA, and this RNA is particularly responsible for the production of variety of the proteins after its translation. So the transcriptional factors and signals go in and out through the nuclear pores under a tight control. Here is another organelle known as endoplasmic reticulum. Number one, rough endoplasmic reticulum. It handles translation and post-translational modification. Protein sorting and vesicles are formed within the Golgi apparatus. So let's have a look on the genetic disorders which are associated. Genetic diseases are often found related to the malfunction in the protein sorting, processing, and trafficking across the organelles. Lysosomes are of cellular digestion, and peroxisomes are responsible for fatty acid oxidation and detoxification. Damages to the lysosomes leading to release of digestive enzyme, and they are responsible for many clinical symptoms. Mitochondria are for respiration and ATP energy production. It has two membranes and its own DNA. They might be originated from the endosymbiosis in primitive eukaryotic cells. So this is the summary. The eukaryotic cell genetic instructions are housed in the nucleus and carried out by the ribosomes. This is structure, nucleus. Here you can see the structure of the nucleus, and this is the endoplasmic reticulum. The nucleus is surrounded by nuclear envelope, which is a double membrane structure perforated by the nuclear pores. These are the nuclear pores. The nuclear envelope is continuous with the endoplasmic reticulum. What is the function of the nucleus? It houses the chromosome made of chromatin, DNA, the genetic material, and protein, and contain nuclei where ribosomal subunits are made. Nucleus also possesses pores. Pores regulate entry and exit of materials in and out of the nucleus. The next organelle is the ribosome. There are two subunits of ribosomal RNA and protein, and it can be free in cytosol and it could be bound to the endoplasmic reticulum. Ribosomes are very much important for the protein synthesis. The next organelle is the endoplasmic reticulum. The endoplasmic reticulum is the extensive network of the membrane-bound tubules and sacs. Membrane separates lumen from cytosol continuous with the nuclear envelope. A smooth endoplasmic reticulum is responsible for the synthesis of lipids, metabolism of carbohydrate, calcium, storage, detoxification of the drugs and poisons. However, rough endoplasmic reticulum, It helps in the synthesis of secretory and other proteins from bound ribosomes and helps carbohydrate to glycoproteins produce new membranes. The other organelle is the Golgi apparatus, which is responsible for the further splicing and sorting of the proteins which are synthesized by the endoplasmic reticulum, which are transported from the rough endoplasmic reticulum. So Golgi apparatus are the stacks of the flat and numerous sacs having polarity, and they are found and they contain cis and trans phases. The modification, they are responsible for the modification of the proteins. They contain carbohydrates and proteins and phospholipids. Synthesis of, they are responsible for the attachment of the carbohydrate within the protein in order to synthesize our glycoprotein. There are this synthesis of many polysaccharides, sorting of Golgi apparatus, which are then released in the vesicles. The next organelle is the lysosome. It is a membranous sac of hydrolytic enzyme in animal cells. It is responsible for the breakdown of ingested substances, cell macromolecules, and damaged cell organelles for recycling. Vacuole, on the other hand, is a large membrane-bounded vesicle in the plant cells. The function of vacuole is the digestion, storage, waste disposal, waste balance, cell growth, and the production of a plant cell. The next organelle is the mitochondrion. Mitochondrion and chloroplast. They change energy from one form to another form. So mitochondria are bounded by double membrane, an inner membrane and an outer membrane. They are responsible for the cellular respiration. Chloroplast, which is present in the plant cell, they are typically they contain two membranes around fluid stroma. These are the stromas. These stacks are the stromas, which contain membranous thylakoid stacked into the grana. They are responsible for the respiration or for the photosynthesis in the plant cells. So the next organelle is the peroxisome, which is a specialized metabolic compartment and bounded by a single membrane. Peroxisome contain enzymes that transfer hydrogen to water, producing hydrogen peroxide as a byproduct, which is then converted to water by other enzymes in the peroxisome. This was the revision of the whole lecture. There was a comparison and a contrast of prokaryotic and eukaryotic cells. Then we compared and contrast the plant and animal cells. Further, the endosymbiotic hypothesis of the origin of the organelles in eukaryotic cells was discussed, and number of organelles and their functions were discussed. Then we discussed what is nucleolus and what is the basic function of the nucleolus. This was all about the cell and its organelles, their function, and the central dogma of life. Thank you very much.